Hello, everybody, and thank you for joining this wonderful session for the Festival for Genomics and Biodata, focusing on the dark targets. My name is Tudor Oprea, and I will be happy to answer your questions if you type them in the chat box, so please don't forget to do that. Just a quick word from our sponsor. This is an NIH-funded project, and it is part of the Illuminating the Druggable Genome project. I am part of what's called the Knowledge Management Center, and in that regard, my focus has been for the past 6 years to gather large-scale datasets and annotate them. So we currently collect information from genome and proteome data, and of course, to process it, we have to bin it in appropriate bins. So sometimes we had to develop our own ontologies, such as the drug target ontology together with Stefan Schürer. And we also observe several disease ontologies, including diseases and monarch disease ontology. We also capture chemocentric data from resources such as Campbell and our own Drug Central, and of course data from literature, patents, drug labels, etc. So all that gets deposited into Target Central, which is then made available either as a direct download, so you can do a complete dump, or you can look at it through this portal, and that's pharos.nih.gov. And if you want to learn more about the IDG initiative, you should visit druggablegenome.net. Just very briefly, these are the data sets that we integrate, those that are highlighted with dark blue, such as Shore Campbell and Harmonizo, are resources that we fund in part. This is, for example, diseases resource maintained by Lars Jensen, and the Harmonizome, which is maintained by Avi Mayan at Mount Sinai, as well as our own Drug Central. We also incorporate data from several other common fund initiatives, such as GTEx, the International Mouse Phenotype Consortium, and LINKS. So all these data get deposited into Pharos, and they are machine learning ready. So if you want to plug and play, you should be able to do so. Part of our work has been to categorize proteins, specifically human proteins, and we proposed what's called a knowledge-based classification, which divides the human proteins into 4 categories. The smallest one, which is at 3 o'clock on the inner ring, is called TCLIN, and these are proteins that are known to be associated as mode of actions for targets. TChem are proteins for which we have an awareness of their chemical annotation. The vast majority are T-bio, which means we understand their biology, but not so much manipulate them from a pharmacological or chemical perspective. And about 1/3 are in T-dark. And those numbers, of course, change over time. How do we get to T-chem? We actually look at all the available drugs that are annotated for that drug category by mode of action. That was subject to a paper in Nature Drug Discovery in 2017, which we continually update. So there's an update on drug targets in Nucleic Acids Research in the January issue, which just got published. And if you look, for example, at the difference in bioactivity spread between kinases and ion channels, you'll see that it is roughly 2 orders of magnitude. The dot illustrates the 90th percentile on the log scale. So roughly 30 nanomolar is the cutoff for kinases, and about 10-20 micromolar would be the cutoff for ion channels. And we therefore use a sliding scale that's based on the different drug targets. When it comes to T-dark and T-bio, I'll focus your attention on T-dark first. We use a fractional publication count, which was first proposed by Lars Jensen. And essentially what we do is we count publications. For example, if you have 100 papers talking about the insulin receptor, and each paper talks only about the insulin receptor, then that count would be 100. But if you have 100 papers talking about 20,000 proteins each, then that count is very, very small indeed. We also look at gene reference into function, which is a resource maintained by NCBI, and antibodies from antibodypedia.com. And all that information gets tagged. If you're not in that category, you end up in TBio, meaning you lack chemical matter, but there are enough publications to describe the target. So all that taken into account, why should we care about the dark genome? Here are some examples that have been annotated and were subject of an NRDD paper in 2018. You look, for example, at the leptin receptor or at PCSK9. Once you had a very clear evaluation of the role, either through the organization or through a strong disease association study, that was enough to prioritize it as a drug target. I would not be surprised if PCSK9 is one of the records in terms of the shortest time from total dark target to drug target, which was roughly 12 years. And on average, it takes about 15 to 20 years to illuminate a drug target. One of the questions that we asked is how can we use additional resources such as the Mouse Phenotype Consortium to address the question, how much do we really know? So this plot, which is categorized by the target development levels, so T-dark, T-bio, etc., asks the question how many of the mouse models that have been phenotyped and evaluated through the IMPC, which as of version 13 of IMPC cataloged about 6,500 human genes. So we look at the orthologs, of course, and about less than 1,000 genes are associated with chemical targets or with chemical probes or drugs. About 3,800 of these are understudied, meaning they have a fractional publication count of below 50, and the majority of them, of course, are T-dark and T-bio. And again, if you want to learn more about Pharos, you go to pharos.nih.gov, and if you look at the top part, you will see several topics that you can search: target, disease, and ligand. And of course, we have an API and information about it, etc. And I would guess the take-home message for this is that I hope I convinced you that we have a knowledge deficit. About 1 in 3 proteins is understudied. That number has been steadily decreasing, and about 3% of the proteome is currently targeted by drugs. That number is very slowly increasing. The increase has been roughly 10% from about 600 to about 660 over a period of 8 years. And also a word of caution, if you try to petition a funding agency and you propose to work on dark genes, that turns out to be a high-risk endeavor. Going then on the question of how to best integrate information through this process, part of the question that I was trying to address is how best can we take advantage of advances in text mining. So the resource that we use is maintained by Lars Jensen in Denmark. It's called Diseases. It's got 3 channels, text mining, knowledge, and experimental. On the experimental side, we recently posted a contribution about a resource called Taiga. So if you're interested in the slides, this will be provided with a link. We provide the rank score of all the GWAS data by introducing additional information such as the relative citation ratio. The neat part about diseases these days is that we've made tremendous advances using machine learning to avoid false negatives when it comes to gene names. I'll just give you an anecdotal example. When we text mined the grants from NIH, so sort of the grantome, if you want, to figure out which genes are mostly funded and which are not funded, the most funded gene was AIM1. And of course, we had to correct for that error. The question then becomes, how do we take advantage of these protein disease associations? It turns out it's a rather complex matter. There's roughly 11 channels that we currently track, including this GENET, which is maintained by Laura Furlong in Barcelona. And of course, diseases, there's several other resources that we take into account. Part of the problem is that all this information is actually being split on a per gene basis, where in reality, most of these proteins make complex associations to form what's called a drug target. This is a simple example where you have an ion channel combining with a transporter to form a sulfonylurea receptor, which is a target for a certain type of anti-diabetic drugs. So unless you know that these have to go together, you will probably not do the proper text mining. This illustrated even further that there really is a translational gap between science and placing drugs, and I choose to focus on rare diseases because I believe that by studying rare diseases, there is a really good opportunity to map that particular disease phenotype to the mouse phenotype. So, for example, when you look at IMPC. But what this graph shows you is that for a period of 30 years, there is roughly a 2 to 3 order of magnitude difference between the number of drugs that enter the market, tracked from the European Medicines Agency and the US FDA, as well as the number of publications which refer specifically to rare diseases and genes. And there is, of course, a lot needed to correct that gap. One way to do that would be, for example, to start with the inner ring. If you look at the T-clean and T-chem, that's roughly 20% of the rare disease proteome. So the possibility to quickly map loss of function in these disease situations with the loss of function caused by a mouse knockout would probably accelerate the process. It's also important to recall that T-clean has the demonstrated benefit that we know these to be mode of action drug targets, which means that at least from a clinical perspective, if you perturb that target, you get a clinical outcome. That is not always the case, as we probably all know. It takes a lot of effort to validate a drug target into the clinic. But nonetheless, there are significant opportunities for those who are interested in this area. This is a plug to the fact that we've recently re-evaluated the number of rare diseases. The most often cited number for rare diseases is about 7,000. That number reflects actually the number of monogenic diseases that were available in OMIM, the online Mendelian inheritance in man database. This was probably the first database to focus on rare diseases, which is roughly a 5-decade effort started by Viktor Krusnik. And what we've done together with Melissa Handel and Chris Mangle and Peter Robinson and the Monarch team was to try to address the question, how many of the rare diseases in the Monarch disease ontology are actually leaf nodes, meaning there are no child nodes in the ontology. The number we came up with is roughly above 10,000, and that's sort of revising the number of rare diseases by upwards of 50% from what was previously believed. And it's also important to know that if rare diseases are not counted, then rare disease patients do not count. We really wanted this to be the title of our publication in Nature Drug Discovery, but the editor thought it was too controversial. But then we can ask the question, how many mouse phenotypes can be mapped to rare diseases? And the answer, looking at the per gene basis, is roughly 2,500. So we have 540 genes that are T-clean and T-chem, and about 1,800 that are T-bio and T-dark. And again, about 1,000 of these are understudied, which means that, say, for example, if you wanted to start with those that are really understudied and go with those, you are more likely to establish very quickly new relationships, and specifically for proteins that lack functional annotation, this might probably give you an idea of how well we can annotate these genes and impute disease associations. So looking at the function, it turns out, for example, if you look at T-dark, about 70% of these are actually proteins of unknown function, and there is clearly a sense of urgency on trying to understand how these map to disease phenotypes that have been observed in humans. And of course, if you follow this dark gray area, you'll see that the majority of them are in the bottom right panel, which is T-dark, and in the upper left panel, which is T-bio. And that's not to be the case for T-clean and T-chem, because those have been very, very well studied genes. Which brings me to protein function or disease imputation. The way we perform that is we implemented a machine learning workflow that starts with scripts that provide the database construction. So on the left top panel, you have the resources that we frequently use for this machine learning. We actually have the option to add additional data sets, but we try to get representatives from across the spectrum, expression data, pathways, protein interactions, etc., etc. So all those get into a model data set creation. So all these get scripted into developing a per phenotype or per disease model, which then is undergoing the metapath transformation. Think of this as the equivalent of data and metadata. So we're looking at a path and then a metapath. So the metapath is literally trying to count how many ways you can go on a knowledge graph from point A to point B. And of course, we give it a label of 1 when we can clearly navigate, say, from the insulin receptor to diabetes, and the label gets to 0 from, say, a completely undescribed protein to a disease. The algorithm that we implemented is XGBoost. We frequently run 100 models. We've recently started to do additional perturbations and look for stability in the models, etc., etc. And then we try to validate the models. Speaking of validation, this is a first example out of the learning curve. So we built a model using the 51 genes from OMIM that are associated with type 2 diabetes. For negative samples, we used the other 4,000 OMIM genes that were not associated with diabetes. So we assume those to be true negatives. This may or may not be correct. But there are not a lot of studies that say this gene is not associated with diabetes. We found, for example, hemoferritin and HMOX1. Both of these are perturbed in a disease called hemochromatosis. Most of these patients migrate to type 2 diabetes, and so we thought this was a reasonable approach. So I sent this information to Mark McCarthy, who before joining Genentech as senior director was leading at the Oxford University a project called Accelerating Medicine's Partnership Type 2 Diabetes Consortium. And as part of this AMP consortium, I thought it would be the best place to try to examine and validate the machine learning model. Turns out, and this is actually the content of an email I got from Mark about 2 years ago, turns out that the training set was not reliable because OMIM is not a causality related database, but it turns out that the model, despite the fact that it indicated some relationships, it wasn't really that informative. So we got a completely different model, much stronger information validation when you look at external results by taking the data from Anubha Mahajan and Mark McCarthy. These are 54 causal transcripts. Once you plug those in, the results are dramatically changed. The two of the top protein-protein interaction nodes are SLC38, is a transporter, and the gastric inhibitory peptide receptor. These have very strong signals associated with diabetes. And in the top 24 genes, that are 9 that are already confirmed by GWAS as being associated with diabetes. And 4 have imputed, not confirmed, coterms for insulin secretion. So overall, a very different model, which brings me to the point that when you do machine learning model, you shouldn't do it in vacuum, but rather talk to experts because they know what they're doing. In addition to that, and the strength for this approach is to look at additional data. So for example, with the IMPC, there are 5 different phenotypes which have at least 60 to 100 genes that are positively associated with that particular phenotype, looking at glucose tolerance and glucose levels in the blood, as well as insulin levels. And then you can run these models in parallel. And on top of that, look at the model with the causal transcripts from Mahajan and McCarthy. And the idea behind this is that you then have a panel of predictions and you can compare them across different multiple models and then prioritize accordingly. So this would be a much more accurate way to predict novel gene associations for a specific disease. So where do we go for dark gene prioritization in precision medicine? One thing that I learned through the example that I showed with type 2 diabetes is that if you apply AI/ML, it doesn't necessarily lead to causality. So practically model quality is limited by the quality of the input data. The old adage, garbage in, garbage out. So you really have to be careful, and it's probably best to talk to disease experts before you build your models. The other observation that's quite relevant is that lacking true negative data can significantly dent the information system. So you're not going to be able to build reliable models without true negatives. In the case of IMPC, because they phenotype all the mice across a battery of 500 plus tests, having no significant phenotype change in that particular test means that if that mouse has been tested but not significantly, then that qualifies as a true negative. So the next step is to combine monogenic rare disease phenotypes with mouse model phenotypes, and that should at least increase our understanding for disease pathology. And of course, there's the caveat of translating mouse knockout data to human biology. But overall, I would say this is a promising rational strategy for prioritizing drug targets. So I guess the second take-home message is that data science and machine learning can definitely help with target prioritization. And to paraphrase William Gibson, from author of The Necromancer, he said this about the future, but the truth is out there is just not that evenly distributed. And any of you who has a lot of expertise in a disease that could benefit from machine learning model, we definitely need your help. As Manolis has indicated earlier, machine learning models can be really rapidly generated, but the really hard part is to validate the experiments. And just as an ending slide, I believe that the dark genome research can benefit the rare disease community. Those of you who are aware, last day of February is Rare Disease Day, so this is an acknowledgement of that, but it's quite clear that both patients and mice can help. And with that, thank you for your attention. I'll be happy to answer questions. What is the biggest scientific surprise from all of the findings from the dark genome research, something that you would have never predicted? So first of all, the biggest surprise for me was the fact that even after 20, 30 years of the Human Genome Project being completed, folks continue to get funded and publish on the same genes that they studied 20 or 30 years ago. This is all published in our landmark publication in Nature Drug Discovery. The other biggest surprise was that if you really think about it, if you take TCLIN and TChem, plus their immediate network, say in string, you end up with about 3,000 proteins. So everything that's in the textbooks of pharmacology and biochemistry refers to about less than 25% of the human proteome. So that to me was mind-boggling, and I would have never predicted it. As far as unlocking new discoveries via machine learning and artificial intelligence, I think there is tremendous potential. The difficulty is literally understanding what you're doing and making sure that you collaborate with clinicians, preferably physicians, scientists, who can speak the molecular biology, but also can help navigate the perils of annotating data. Also, a lot of people talk mining data from like real-world evidence, so trying to mine data from patient records. Those are also fraught with dangers. The simplest case in point is just look at the birthdays or the patient visit date. Some of them go straight to the future, like 2050. So if that level of annotation for birthday or date of the visit is there, then you also have to question the level of annotation about just everything else that's in the database. So machine learning can help, but it's only as good as the data that you provided. The next question is, when, if ever, do you think we will have a complete understanding of the entire genome, the function of every gene, and the influence of genes or sequences that do not code for a protein? The Guardian today has a story, could you do something with $1 trillion and solve all the problems of all diseases? So I think if we get enough funding and enough people to work on this, we could accomplish this task in 10 years. But if we keep trickling down the funding, it may take 20 to 50 years. It really depends on our ability to generate the proper experimental setups, validating separately and independently the same information. So make sure we're not on a wild goose chase. And I think it's possible and doable and within reach. The next question, is there a second order T-dark, genes that are absent from the standard reference, or is presence absence now well annotated? The answer is no. We rely on UniProt and the human curation of proteins. What we found over the space of the last 8 years, we have an additional label, which we call T-void. T-void are proteins that have retired because they turned out to be pseudogenes, or they just appeared because somebody has added them. So from T-void, it migrates somewhere else. But the shorter version is there's about 2,500 human proteins that have absolutely no information whatsoever. The next question, how would you compare your efforts establishing gene disease associations to those of open targets in the UK? These are parallel efforts. We run them. We often talk. In fact, we're in the process of hiring a joint postdoc that will be hosted by Open Targets, but it will be half funded by NIH. And the task of that postdoc will be to help us improve the way we prioritize targets for drug discovery. So I think the efforts are in parallel, and there is, of course, the inevitable duplication, but there are unique features in both that they play to their strengths. I don't know if there are any other questions, but with that, thank you everyone for your attention, and feel free to contact me via email at toprea@salud.unm.edu. Thank you.